Welcome everyone, myself Rohan Zaveri from the team Antidote, and here are our other team members. Today we will be presenting the topic microfluidic devices. We will try to delve into the briefs of their fabrication techniques, characteristics, components, etc. To introduce you to the topic, let's see the 1990s computers. They used to take up space of a whole room to store just a few MB of data. And these are today's chip-sized computers, which can store tons of data at your fingertips. This pursuit of miniaturization in chemical engineering and biotech has led to this microfluidic technology, which is poised as one of the biggest technological advancement that will lead the upcoming generations forward. So what is this technology? Microfluidics is a technology to manipulate or process small volume of fluids using microchannels that range from tens to hundreds of micrometers. As you can see in this chip, these dimensions are in picometers. But the obvious question that arises here is, why do we need them? So the answer lies in their characteristics. The first is the low sample volume. Obviously, you cannot use liters of bloods to diagnose for something. That's why we need chips which are so small in size that you just need a drop of blood. Classic example is this device in the image, or you have seen the COVID RT-PCR test. Second is the scaling out. Parallel structures allow multiple reactions in small area, so we can perform multiple reactions as well on the same chip. And the third one is the shorter reaction time. These chips are very quick, affordable and very scalable.
Here are some key examples. Biomedical diagnostics. Microfluidic devices enable rapid, low-cost diagnostic testing by analyzing small fluid samples. For instance, Abbott's ID Now provides fast molecular testing for diseases like COVID-19, which was widely used during the global pandemic. While lab-on-a-chip (LOC) devices can detect multiple medical conditions from just a few drops of blood. Environmental monitoring. Microfluidic sensors are used to detect pollutants and contaminants in water, air, and soil. These devices offer quick and accurate monitoring of environmental toxins, heavy metals, and pesticides, helping ensure public health and safety. Drug discovery and development. In pharmaceuticals, microfluidics accelerates drug testing by enabling high throughput screening in controlled environments. Aiche, a company now part of Merck, uses microfluidic technology for testing drug responses in living cells, streamlining the early stages of drug development, food safety, and quality control. Microfluidic devices are used to detect allergens, pathogens, and other contaminants in food products. Companies like Sigma use microfluidics to test for food allergens like peanuts or gluten directly at production sites, ensuring food safety. Energy generation and fuel cells. In the energy sector, microfluidics improves the performance of microfuel cells by optimizing fuel flow and reaction efficiency. Many companies manufacture hydrogen-based fuel cells for remote power generation using microfluidic technology to enhance the device's performance.
Let us now see how microfluidic devices are fabricated. The first step is the selection of material. Durability, transparency, ease of fabrication, no reaction with analyte, and low cost are a few properties on the basis of what the materials are assessed. Typical materials which are used often are glass, silicon, and polymers such as PDMS, which is polydimethylsiloxane. Now let us see the fabrication techniques. Microfabrication methods include lamination by layer bonding, soft lithography by PDMS molding, and 3D printing. The first one, which is lamination. It involves cutting, aligning, and bonding multiple layers like plastic or glass. This layered approach enables precision and flexibility in microfluidic channel design. The second one is soft lithography. It uses photolithography to create a master mold, which is then cast with PDMS to produce microstructures. and this process is ideal for replicating complex micro patterns. The last one, which is 3D printing, it is a process in which the fused deposition modeling takes place, in which thermoplastic materials are melted layer by layer. It is affordable, uses biocompatible plastics, but it may be leak prone for microfluidic substances or devices.
Let us now understand fluid dynamics at microscale, how it is different from macroscale. Fluid dynamics in microchannels is governed by principles that differ from those at macroscale due to the small dimension and dominance of forces like viscosity, surface tension, and diffusion. In contrast, macroscale systems are often dominated by interstitial forces, convection, and turbulence. First is the laminar flow. Due to very small diameter of channels and less velocity, the flow is highly laminar and Reynolds number is much less than 1. Thus in such a cases, mixing of two streams is very difficult to achieve. So mixing at microscale is dominated predominantly by diffusion, which is very slow. Thus we need to apply various techniques to increase the mixing. Unlike at macroscale, we can use aggregators, stirrers, etc., which are not feasible and applicable such as microscale.
Efficient mixing is very important for any purpose of microfluidic device. Therefore, there is a need to increase the mixing efficiency by employing various techniques. There are two basic methods for increasing mixing: passive and active. Passive mixing elements are commonly incorporated into microfluidic devices during fabrication. As the name suggests, these types of elements are passive and they do not require any additional controls by users. They are often achieved by utilizing clever alteration to the channel walls, geometry, or surface properties. The core principle behind the passive mixing is to enhance molecular diffusion by increasing the contact area or contact time. All the passive mixing techniques revolves around using these principles. The most used is split and recombination of streams, in which stream is divided into confluent streams and then further recombined. In contrast to passive mixing enhancement, active mixing enhancement offers more flexibility in the degree of mixing achieved. However, due to tiny moving elements and such a precise control demands much more elaborate and expensive production. The most widely used is the electroosmosis. It employs electrodes inserted in a specific channel to create of an electrostatic field that drives the neutral liquid contained in it in the direction of the electric field, increasing the velocity profile and thus aiding in mixing. That's all for the today's presentation, guys. We hope we made you aware about this upcoming emerging field and how it can impact the future market. I thank you very much for your patience in listening us.